Cancer cells frequently employ a devastating immune evasion strategy, the downregulation or complete loss of Major Histocompatibility Complex Class I expression. This mechanism prevents cytotoxic T lymphocytes from recognizing the tumor. Normally, T cells rely on the T cell receptor binding to specific peptide antigens presented by Major Histocompatibility Complex molecules on the cell surface. When Major Histocompatibility Complex Class I is absent, this crucial recognition pathway fails. Furthermore, many tumors lack highly specific immunogenic antigens recognizable by endogenous T cells. This failure of Major Histocompatibility Complex restricted recognition necessitates an alternative targeting mechanism. Chimeric Antigen Receptor T-cell therapy is designed precisely to bypass the requirement for Major Histocompatibility presentation entirely, enabling T-cells to recognize tumor-specific surface proteins directly, regardless of the cell's Major Histocompatibility Complex status. A chimeric antigen receptor, or CAR, is a synthetic receptor engineered to redirect T-cell specificity. The CAR structure is modular, consisting of three primary functional domains. The extracellular domain is typically a single-chain variable fragment, abbreviated scFv, derived from a monoclonal antibody. This scFv is solely responsible for binding the specific tumor-associated antigen. The scFv is linked via a hinge and a transmembrane domain to the intracellular signaling components. The core activation signal, known as signal 1, is provided by the cytoplasmic tail of the CD3 zeta chain. Upon antigen binding, the clustering of CARs initiates T-cell activation through this CD3 zeta domain. This modular design allows the engineered T-cell to function as a targeted killer completely independent of the Major Histocompatibility Complex presentation system. Chimeric Antigen Receptor T-cell efficacy is fundamentally defined by the inclusion of costimulatory domains, which categorize the CARs into generations. First-generation CARs contained only the CD3 zeta signaling domain, resulting in limited T-cell proliferation and poor persistence in the patient. Second-generation CARs incorporate one costimulatory domain, typically either CD28 or 41BB, also known as CD137, along with CD3 zeta. This addition provides signal 2, significantly enhancing T-cell activation, cytokine production, and long-term persistence. Third-generation CARs include two distinct costimulatory domains, such as CD28 and 41BB, aiming for maximal signaling output, though clinical superiority over second-generation designs remains context-dependent. Fourth-generation CARs, often called T-cells redirected for universal cytokine killing, are engineered to secrete additional factors like interleukin-12 to actively modify the immunosuppressive tumor microenvironment. Chimeric antigen receptor T-cell manufacturing begins with leukapheresis, a procedure collecting peripheral blood mononuclear cells directly from the patient in the autologous approach. T-cells are subsequently isolated and activated ex vivo, typically using anti-CD3 and anti-CD28 antibodies or specialized beads. The crucial step involves introducing the CAR gene into the T-cells, most commonly achieved via viral transduction. Replication-incompatible lentiviruses or retroviruses are used as vectors to deliver the CAR construct DNA into the T-cell genome, ensuring stable, long-term expression. Following successful transduction, the engineered T-cells are expanded exponentially in specialized bioreactors over several weeks to achieve the required clinical dose, often billions of cells. The final product is then cryopreserved and subjected to rigorous quality control testing before being infused back into the patient. Before infusion, patients typically undergo lymphodepleting chemotherapy, usually involving fludarabine and cyclophosphamide, to reduce endogenous lymphocyte counts. This step is essential to enhance chimeric antigen receptor T-cell engraftment and persistence. Once infused, CAR T-cells circulate and bind to the target antigen on the tumor cell surface, exclusively via the single-chain variable fragment domain. This binding event triggers the clustering of the chimeric antigen receptors and the subsequent activation of the intracellular CD3 zeta and costimulatory domains. The activated CAR T-cell initiates cytotoxicity by rapidly releasing lytogranules containing perforin and granzymes. Perforin creates pores in the target cell membrane, allowing granzymes to enter the cytoplasm. Granzymes then induce caspase-mediated apoptosis, leading to rapid and targeted tumor cell destruction. Chimeric Antigen Receptor T-cell therapy has demonstrated profound efficacy primarily in hematologic malignancies. The most successful clinical target is CD19, a B-cell surface marker used to treat refractory B-cell acute lymphoblastic leukemia and aggressive B-cell non-Hodgkin lymphomas. Anti-CD19 CAR T-cells achieve complete remission rates exceeding 80% in pediatric and young adult acute lymphoblastic leukemia patients who have failed standard therapies. B-cell maturation antigen, or BCMA, is the primary target for treating multiple myeloma, showing deep and durable responses in heavily pretreated patients. This clinical success relies critically on two factors: the uniform expression of these antigens on malignant cells and their restricted expression on non-essential healthy tissues, specifically mature B cells and plasma cells. The rapid activation and massive proliferation of chimeric antigen receptor T-cells can induce severe systemic toxicities. Cytokine Release Syndrome, abbreviated CRS, is the most common adverse event, resulting from the massive release of pro-inflammatory cytokines. These include interleukin-6, interferon gamma, and tumor necrosis factor alpha, released by activated T-cells and bystander immune cells. CRS symptoms range widely, from high fever and hypotension to potentially fatal multi-organ failure. Management involves supportive care and the targeted interleukin-6 receptor antagonist tocilizumab. Immune effector cell-associated neurotoxicity syndrome, or ICANS, is another serious toxicity characterized by symptoms like confusion, seizures, and cerebral edema, requiring aggressive corticosteroid intervention in severe cases to mitigate central nervous system inflammation. Current research focuses intensely on overcoming limitations, primarily the poor performance in solid tumors. Solid tumors present major challenges, including significant antigen heterogeneity, poor chimeric antigen receptor trafficking into the tumor mass, and the highly immunosuppressive tumor microenvironment. This environment is rich in inhibitory cells and checkpoint ligands. Strategies to address the tumor microenvironment include armored CARs engineered to secrete activating cytokines like interleukin-12 or express dominant negative receptors to block inhibitory signals. Allogeneic CAR T-cells derived from healthy donors offer an off-the-shelf alternative to the costly autologous process. Allogeneic approaches require precise gene editing, typically using CRISPR-Cas9, to knock out the endogenous T-cell receptor and Major Histocompatibility Complex Class I molecules to prevent graft versus host disease and host rejection, respectively.